Hello and welcome to another tutorial for BIOS E1A. In this tutorial we'll walk through a practice problem dealing with the material covered in the lectures on Mendelian genetics. Let's go ahead and get right to the question. First, we should unpack the given information in the intro paragraph of the problem. What exactly do we know? Well, they tell us that we're dealing with guinea pigs as the organism we're considering. We're also told that we're dealing with two different genes in this problem, one for the guinea pig's coat color, that is the color of their fur, and the other for the length of their hair. They also tell us that the two genes assort independently, meaning that they are on different chromosomes and that we can avoid thinking about linkage in some of the more complicated scenarios that we encountered in the second Mendelian genetics lecture. For the coat color gene, we know that the dark coat color is dominant over the albino coat color. What that means is that there are two versions or alleles of the coat color gene, and that the dark coat color allele is the dominant allele, represented by the capital C, and is dominant to the recessive albino coat color allele, represented by the lowercase c. Guinea pigs that have two big C dominant alleles will have dark coat color. So will guinea pigs that are heterozygous for the coat color gene, meaning that they have one big C dark coat allele and one little c albino coat allele. They'll have dark coats because the big C dark coat allele is dominant. Only guinea pigs that are homozygous for the recessive lowercase c allele, that is, that have both copies being the recessive little c albino allele, will have albino coats. The introductory paragraph gives us the same information about the hair length gene. Short hair is dominant over long hair in guinea pigs, and there are two alleles for the hair length gene: the dominant allele for short hair, represented by capital S, and the recessive allele for long hair, represented by the lowercase s. Homozygous big S guinea pigs, or heterozygous big S little s guinea pigs, will have short hair, because they have at least one copy of the dominant allele. Only the small s guinea pigs, homozygous for the recessive allele, will have long hair. So that's the setup information that they give us for the problem. Before we dive into solving parts A through G, let's step back and review what exactly happens for a single gene when you know the genotype of both parents. More specifically, how do we predict what percentage or fraction of their offspring will have which phenotypes and which genotypes? The way we've seen to work on this in the course lecture is by using Punnett squares. Let's review how that works. As an example, let's consider the scenario where you have two parent guinea pigs, each of which is heterozygous for the coat color gene. That means each parent has two different copies of the gene. One copy is the dominant big C dark coat color allele. The other copy is the recessive little c albino coat color allele. When those two guinea pigs mate, what fraction of their progeny will have what phenotype? We can set up a 2x2 Punnett square to answer this question. First, make a 2x2 grid. Then on one side, put the possible gametes that one of the parents could produce. Parent 1 is a heterozygote here, meaning their diploid cells will contain both a dominant big C allele and a recessive little c allele of the coat color gene. Parent 1's gametes, though, will be haploid, and each gamete will only have one copy of the gene, either the big C dark coat allele or the little c albino coat allele. The same thing is true for parent 2 in our scenario here. Once you've put the possible gamete genotypes on the rows and columns on the sides, you can combine them to fill out the Punnett square table, like so, giving you all the possible genotypes of the progeny from this cross. By looking at what phenotypes these genotypes will generate in the guinea pig offspring, you can then compute what proportion of the progeny from the cross will have what phenotype. In other words, you can get an expected phenotype ratio for the progeny that would result from breeding these parent guinea pigs together. Remember that big C guinea pigs and big C little c guinea pigs will have dark coats, that is, the dominant phenotype. They both have a copy of the dominant allele. Only the little c recessive homozygotes will have the recessive phenotype, an albino coat color. In this scenario here, where both the parents were heterozygous, 3 out of 4 progeny will have the dominant dark coat phenotype. Only 1 out of 4 will have the recessive phenotype, albino coat color. This gives a 3-to-1 phenotype ratio. This ratio is characteristic of a single gene cross where both parents are heterozygous. Recognizing this 3-to-1 ratio is key to solving this practice problem and other ones like it. Let's look at a couple more phenotype ratios that you get when considering a cross involving a single gene. Another important type of cross you heard about in lecture is the test cross, the cross where one of the parents is homozygous for the recessive allele. Again, using guinea pig coat color as an example here, This would mean that one parent has the recessive little c allele for both their copies of the coat color gene, meaning that all the eggs or sperm that they would make carry the little c albino allele. Let's fill that part into some Punnett squares here. With the test cross, there are three possibilities depending on what the other parent's genotype is. If the other parent is also homozygous recessive, then all the progeny must receive only recessive alleles. We can see that here with our example with the guinea pig coat color gene. Two homozygous recessive parents are bred together, and the result is that 100% of the resulting progeny will be little c, homozygous recessive with albino coat color. Another test cross outcome would be that the homozygous recessive parent 1 is bred with a heterozygous parent 2, as is shown here. In this case, when you fill in the Punnett square, you can see that 50% of the progeny will also be heterozygotes, and 50% will be homozygous recessive. In our example here, that means half of the progeny are big C, little c, and will have the dominant dark coat color. Half the progeny will be little c, and have the recessive albino coat phenotype. This yields a characteristic 1-to-1 phenotypic ratio. Finally, you could also have the second parent being homozygous for the dominant allele. In that case, all the progeny, 100%, will have the dominant phenotype. In fact, this is generalizable for our simple example in this practice problem with a single gene with one fully dominant allele and one recessive allele. If one parent is homozygous dominant, all the gametes it will be able to make will have to carry the dominant allele. So in our guinea pig example with the coat color gene, if one parent is homozygous big C, every gamete it will make ever will carry the big C allele. So no matter whether it is mated with a big C, big C little c, or little c second parent guinea pig, 100% of its progeny will have the dominant dark coat phenotype because they'll always have received or inherited a big C allele from their homozygous dominant parent. These are the possibilities for phenotype ratios in progeny for the basic scenario like we have in this practice problem, with two alleles, one dominant, one recessive, for one gene, diagrammed out like this on a 2x2 Punnett square. If I've belabored this a little bit, that's because it's absolutely critical that you both understand and also really deeply ingrain these different possibilities in your mind if you want to successfully work through the problems on Mendelian genetics like this practice problem, which you will encounter this course.